Hello, my name is Laney Buchanan, and welcome to my poster presentation. The title of my presentation is Engineering Radiopack Materials for Biomedical Devices. So, why use radiopack materials in biomedical devices? Well, radiologists use clinical imaging like computed tomography, magnetic resonance imaging, or ultrasounds to check the location and integrity of the biomedical devices. However, most devices, once implanted, are difficult to distinguish from native tissues. Adding contrast agents like tantalum oxide to these devices significantly improves radiologist evaluation of the implant location, type, and damage. Figure 1 shows that devices that incorporated tantalum oxide performed way better than ones that did not. So, to set up a 20-week degradation study, 3 different polymer matrices had to be made into scaffolds that mimicked biomedical devices. The 3 different FDA-approved biocompatible polymers used were poly-E caprolactone, or PCL, 50/50 polylactide coglycolide, PLGA, or 85/15 polylactide coglycolide. And then 4 different weight percentages, 0%, 5%, 20%, and 40% of tantalum oxide were incorporated into these scaffolds. The scaffolds were individually placed in 1.5 milliliter Eppendorf tubes and were hydrated with either phosphate buffered saline with 0.2% sodium azide, sodium citrate buffer pH 6.5, or sodium citrate buffer pH 5.5. Each week, the supernatant was changed out to maintain sink conditions, mechanical properties, mass loss, and radiopacity were recorded. Data was also collected and analyzed using micro-CT scans. Over a 4-week period, it was apparent that some of the samples changed and some of them stayed the same. Figure 3A and 3B show that the PLGA 50/50 40% tantalum oxide scaffolds had mass and diameter loss, as well as became denser and deformed more easily after mechanical testing was done. Figure 3C and 3D show that having a contrast agent incorporated into these devices makes a huge difference in how they show up during imaging. Figure 4 shows that degradation was impacted by polymer type and weight percent of tantalum oxide. In the figure, it is apparent that the PLGA 50/50 20 weight percent tantalum oxide degrades the fastest and loses its form, and the PCL 20 weight percent tantalum oxide degrades the slowest. The goal of this study was to get a baseline prediction of how biomedical devices would interact with the human body if tantalum oxide nanoparticles were incorporated. Overall, we have shown that degradation is significantly affected by weight percent of tantalum oxide and polymer type. Future studies will focus on device characteristics during in vivo studies with CT visible contrast agents.